The overall goal of this procedure is to identify HIV-positive cells using an HIV-1 Rev-dependent lentiviral vector carrying the green fluorescent protein. This is accomplished by first producing the Rev-dependent lentiviral particles by co-transfection. The second step of the procedure is to infect human CD4 T cells with HIV-1. Next, the HIV-positive human CD4 T cells are superinfected with the lentiviral particles carrying the GFP gene. The final step is to identify HIV-positive cells by measuring GFP-positive cells by flow cytometry. Ultimately, results can be obtained that show the percentage of GFP-positive cells through flow cytometry.
The implications of this technique extend toward therapy of HIV infection, due to the fact that the Rev-dependent vector can also deliver and selectively express therapeutic genes in HIV-infected cells to induce killing of HIV-positive cells. Demonstrating the procedure will be Jia Guo, a research assistant professor from our laboratory.
To produce Rev-dependent lentiviral particles, HEK293 T cells are transfected with three plasmids: the Rev-dependent GFP lentiviral vector, an HIV-1 packaging construct, and a plasmid carrying the VSV-G glycoprotein. 1 day prior to plasmid transfection, seed 2 × 10⁶ HEK293 T cells in 10 ml of medium in a 100 mm Petri dish. Grow cells overnight at 37 degrees Celsius, 5% carbon dioxide. On the following day, replace the supernatant from the cells with 5 ml of fresh medium, then continue to culture the cells for 4 hours at 37 degrees Celsius, 5%.